Hello, everyone.
Hello.
Hello, John. How are you?
I'm good. How are you? A little
I'm
sleepy this
okay.
morning.
Oh, are you tired?
Yeah, it's my first good night of sleep in like a week or so due to one of the kids being sick.
Oh, no.
So I wake up groggy, I guess. Hi, Thomas.
Hi guys, how are you? Good to see
Good.
you.
Hello, Thomas. Thank you for joining the Journal Club and accepting the invitation.
Yeah, thank you so much. That's a big honor.
We're waiting for Keri to join. I think she will do shortly, but we can do a check with you to see that everything works in your presentation.
Yeah, let's do that.
Since you mentioned you had no preference, I was thinking for Keri to go first, and you can have the second presentation, if that's okay.
Yeah, sure, no problem.
Okay.
So should I try sharing my screen?
Yes, definitely.
Alrighty. So I'm gonna do that. And here we go. How does it look?
It's fine.
Everything
It's full
okay?
screen. Mm-hmm.
Yeah. It's changing.
Yes, it is.
Perfect. Okay, well, I guess
Okay.
that's it. I don't have any sounds, so that should
Okay.
be fine.
That was my next question. If you had any.
Yeah. No, I didn't want to risk that. So
Okay.
no, it's all just pictures. So if this is fine, I suppose. Yeah. Okay. Well,
Okay, cool. Yeah.
easy.
Yes. I must tell you that my music instructor is waiting for your talk.
Oh la la. Wow.
Yes.
I hope I won't disappoint.
Actually, he's not a researcher, but he actually wrote a book from a musician's perspective on how music is formed in the brain. So that's why he started looking into it right now, and he's really excited.
Oh, okay. That's super cool. We are organizing a conference with Rick Cohn and Jose Oliveira in Coimbra in October, and there's going to be like a lot of musicians and composers, but also scientists.
Okay.
It's
Have you
about
shared,
like musical time.
have you shared with us the link or the advert?
Yeah. I think yeah. I think Rick reached out to TRF, so I think it must have been in one of the newsletters.
I might have missed it because
no.
we are preparing, me and another two people, we are preparing the newsletter, but we have split it the parts. But yeah, that sounds interesting.
Yeah. I'm curious. It's always nice to exchange with people who are actually like doing the thing, rather than just studying the
Studying, yes. But I think what usually motivates us is what we're interested in general. So we start searching and doing research on it. So that's why we have a lot of musicians in the community.
Yeah, absolutely.
Tomas, do you compose or play much?
It's been a long time, but no, I used to play drums quite seriously. But yeah, since I started the PhD, it was less and less. But I know do you play the violin?
Yeah, I'm a fiddler and guitarist.
Yeah.
Yes, we can.
So do you still have time for music or?
A little. I've got 2 kids now too, but I'm get into Toronto for occasional jam sessions, traditional music.
Oh, that's cool.
And here I've been in between meetings. I'm experimenting with the kalimba tar, where I clamped a kalimba onto my guitar, and I'm trying to figure out how to play them both at the same time.
Okay. Wow.
It's been done before. I'm not the first.
All right, I have to Google this.
There's one person in particular. Let me send a link.
All right. There should be an interim performance between the 2 talks.
Oh yeah. Definitely.
Oh, there you go.
Or
I'm at the very early
we
stage,
can have it. Maybe in the next year, if John, you would probably join the next TRF conference, so we can have a jam session there, and you can play
that would be lovely.
Violin and guitar.
Mm. Are you guys? Sorry.
I was just gonna say, I when I was in 5th grade, I wanted to play the drums, and you had to audition to play the drums in the student band, and I didn't make the audition, and so I fell back on violin. So I think I'm the exception that proves the rule that all the rhythm researchers are percussionists.
Yeah. But I think deep down you're still a percussionist.
Still, I play like a drummer.
Yeah. I want to ask if you guys are planning to go to RPPW in 2 weeks, or
I will be there.
Oh, excellent. Okay.
No,
Sometimes they do jam sessions. Oh
yeah. I hadn't yet thought about whether I'm bringing an instrument. I need to bring an instrument, I guess.
I don't know how much Darren's gonna provide. Perhaps he has everything.
Yeah, you never play somebody else's violin. That's bad hygiene.
True. This is a bit different. Yeah.
Hello,
Hi,
Keri.
Keri.
Hi, how are y'all doing?
We're okay. How are you? Thank you for joining.
Good. I'm good. Just getting settled in Florida.
Oh yes, you're starting at your new role. Good luck.
Yeah, tomorrow actually.
Oh, it will be fine.
Yeah. Everything's still in boxes. Just got my stuff like 5 days ago, so I'm trying to get situated.
Yeah, it will take a little bit, but I'm sure you're gonna settle in. Okay.
Yeah.
So thank you twice for joining today. Then, since you currently moving house.
Yeah, I'm making do.
We already did the tech check with Tomas. So if you can share your screen and see that if everything works. Okay.
Sounds good. Let me start my slideshow. Oh, yeah. Okay. Let's do it this way. All right. Everything pure okay?
Yes, it's full screen.
Awesome.
Try and change your slides also.
Yep.
Yeah.
Seems to be good.
Do you have any sounds that you want to test?
Yeah, it's only one, but we can, it's not detrimental, but we can check it. Just to see. Let's see. Oh, click too fast. All right. Yeah, that's the only sound I have. So if you're able to hear that, we should be good.
I saw the video, but I didn't.
You didn't hear an auditory tone?
Sound? No.
Okay.
I don't know if somebody else heard something.
No.
Not sure.
It's not the end of the world.
No, it's not.
It's an auditory tone. We know what they sound
perhaps like. There is a Keri, perhaps there is a setting that you have to like click something like share sound when you're sharing the screen. This is on Zoom or Teams. I'm getting it mixed up now, but
Let me see.
Maybe
Stop
there was
sharing.
this. Yeah.
Let's look in settings.
Yeah. So when you click, hang on. Oh no, it's not there. It's only when you're.
Yes. Let's see.
In the bottom left-hand corner of the share screen menu, you can see share sound.
Okay. Ah, share sound. Okay, got it. Now let's see if we hear it.
There's a
Yes.
tone.
Cool. Problem
There we
solved.
go. Yes. Nice. I suggested if you're up for it, you can go first, Keri,
Sure.
and then Thomas can go second. Do you prefer the questions at the end of your talks, or do you like people interrupting in between?
Interrupting is fine. I don't mind.
Okay. I will do a brief introduction and mention some ground rules. We usually say that they can raise their hand at the end or place the question at the chat button. But
Okay.
feel free to, when you start your talk, you can mention that you don't mind either way
Okay.
having questions.
Sounds good.
Nice. Okay. We have another 4 minutes left for people to join in. I can see we have here a few participants already, but we will give a few more minutes.
Hi, Martin.
Hi, Keri.
Hello, Martin.
Hi, Effie. How are you?
I'm okay. How are you?
Good. I don't know if you want to say anything about TRF3 before we start, or the only thing we can say is just a reminder that registration, early bird registration goes until June 16th, and
Okay.
just
Okay. I will mention that.
Okay.
Okay, I think it's time to start, and we will have more participants coming in soon. So, hello everyone to another journal club. We are pleased to host two talented postdoctoral fellows today, Dr. Keri and Gladhill and Dr. Tomas Lenc. But before we start, just a quick reminder. The early bird registration for TRF3 goes up to June 16th, so make sure you register soon. And also a quick reminder about our ground rules, as we do every time. Please take your microphones off. You can place your questions at the chat button or raise your virtual hand, and I can read the questions out loud for you at the end of each talk, but based on the preference of the speaker, you can interrupt in between. We would like to thank Dr. Weiner, Dr. Vadakis, Dr. Teki, and the ACR committee for putting these meetings in place. And I think we are ready to start. The first speaker for today, as I said, is Dr. Keri Gladhill. She received her bachelor degree in psychology from Sippenburg University in Pennsylvania in 2016, followed by a master's degree in experimental psychology at St. Joseph University in Philadelphia in 2018. She just completed her PhD in psychology with a concentration in cognitive neuroscience from George Mason University in Fairfax in Virginia, and shortly she's going to work as a postdoctoral fellow at Florida State University. The title of her talk today is The Effects of Movement Parameters on Time Perception. So, Keri, feel free to start. The floor is yours.
All right, let me get my screen pulled up and we can get started. Okay, everybody can hear me okay? See my screen?
Yes.
Okay. All right, well, hi everybody. Like I said, my name is Keri Gladhill. I just finished working at George Mason University with Dr. Martin Weiner, studying time perception, specifically all about how movements affect time perception. For the sake of time today, I'm just going to be talking about one of the studies as part of my dissertation. So I'm sure we're all familiar with these two classic time perception paradigms, the temporal reproduction task on the left and the temporal bisection task on the right. The important thing to note about these two different paradigms is that temporal reproduction is considered motor timing, since motor action determines the to be timed interval. So participants have to push a button on time to time an interval. Whereas a temporal bisection task, there's no overtly timed movement necessary. So they're just simply responding, in this case, saying whether the duration was short or long. What's important to note also is that the areas of the brain traditionally thought to be involved in motor control are active in both of these situations. The main brain area we're talking about here is the supplementary motor area, which we have shown found that it has been chronotopically organized in a rostral-caudal gradient, and that the more rostral neurons encode shorter durations, whereas more caudal neurons encode longer durations. And a study that we performed in Dr. Martin Weiner's lab, in which participants either estimated and reproduced either time or distance during in a virtual environment during an fMRI, we found the SMA to be exclusively activated for coordinating movements for time, but not distance. There's additional evidence for the role of the SMA in EEG studies, specifically the event-related potential known as the contingent negative variation, or the CNV. And we found that has been shown that the CNV amplitude is more negative going when longer durations are perceived. One example of this is a study that I did earlier in my PhD, looking at emotion and time perception. And participants saw a face on either side of a temporal stimulus, and these faces could fall in one of three orders, conditional orders. So they could see a neutral face, then a neutral face, then a negative face, or a negative face, then a neutral face. What we found is when participants saw a negative face before the temporal stimulus, they perceived that temporal stimulus as longer. And when we looked at the CNV, we saw that there was a more negative going amplitude in that condition as well. So it does seem to correspond with perceived duration and is thought to originate from the supplementary motor area. So there's a main theory about how it is that we go about learning to time, and this is done by work from Joyt-Vallée and Kuhl, in which they found that children's explicit duration of estimates are biased. However, when the time is measured implicitly through reaction time, the representation of time is no different than that of an adult. Therefore, it may not be that children are unable to perceive durations, but may not have the cognitive resources to turn that perception into an explicit abstract representation of time. So they went on to elaborate on these findings through a review that was published in Trends in Cognitive Science, in which they suggest that actions aid in the transfer from implicit to explicit representations of duration, and that our ability to understand and use time as adults develop through childhood by sensory motor experiences in the world. If this is the case, this could help explain the involvement of motor areas such as the SMA and time perception, the use of spatial terminology to describe time, such as this is a long presentation, and why movements and time just seem to be so intrinsically connected. The problem remains, however, that classic paradigms and models of timing do not take movements into consideration, and instead they have been considered the endpoint of a time process with motor variance as a nuisance to be partialed out. So to address this issue, we published a review in Trends in Cognitive Science all about how movements shape the perception of time. In this review, we go over a few main effects that movement has on time perception, as well as propose a Q combination framework to explain the differential effects. So I'll summarize a little bit of that now. So one of the main effects is that concurrent movements can bias time perception, in this case, the accuracy of timing. One example of this is by Yokosaka and colleagues, in which participants performed circular hand movements at different speeds. They found that faster hand movements led to a compression of time perception. Another example is research done by DeCock and colleagues as part of this ongoing collaboration, in which increased viscosity of movements led to a compression in time. So participants were using a robotic manipulandum and moving around during a temporal interval and experienced different viscosities of that robotic arm while moving. And this was found compression in both bisection and reproduction tasks. We suggest that these effects of concurrent movements biasing time perception is actually an influence of magnitude. So we know that different magnitudes of numerosity, size, length, et cetera, affects time perception. And so it wouldn't be surprising if different movement statistics also bias time perception, because we intrinsically know short, fast, and near movements take less time, and long, slow, and far movements take more time. The other main effect is concurrent movements improving time perception. In this case, we're talking about improving the precision of time. So one example of this is research done by Martin Wiener and colleagues, in which they had participants do an auditory temporal bisection task. Again, this is using the robotic manipulandum, and participants either held still during an auditory interval or were able to freely move the robotic handle around. And what they found is that there was an increased precision in time perception in the free movement condition compared to the hold condition. You can see in this figure down here, but there was no effect on accuracy or the bisection point. We therefore suggest that movement actually serves as an additional channel of temporal information that's low in variability and high in temporal fidelity, which is leading to these improvements in precision. This brings us to the Bayesian Q combination framework we propose to explain these two different effects of biasing and improving the precision of timing. And this is based on Bayesian decision theory, in which sensory estimates are drawn from a likelihood function with a specific mean and standard deviation. And they're combined with prior distributions, the prior distribution based on learned experiences, in order to form a posterior estimate that is more precise. However, when there's multiple sensory modalities present, maximum likelihood estimation comes into play, and the multisensory estimate is drawn from the likelihood function, which is determined by a weighted sum of the unisensory estimates. And the mean of that multisensory estimate will be closer to the unisensory estimate of whichever modality is more precise. For example, when in timing we have, the auditory is more precise than visual timing. So if they're experiencing a temporal interval, both in auditory and visual modalities, the multisensory estimate of those together will be more precise than either unisensory alone, but it'll be drawn towards auditory since it is the more precise modality. Therefore, we suggest, as I said, with movement, if movement serves as an additional channel of temporal information that's low in variability and high in temporal fidelity, we should see this improvement as the multisensory estimate should be pulled toward that more precise unisensory estimate. But it can also be biased by movement statistics such as length, distance, et cetera, leading to this biasing effect. So we went on to test this framework. This has been part of an ongoing collaboration between Dr. Wilson Joyner at UC Davis and Dr. Martin Weiner at George Mason University. On the left at George Mason University, we have this kind of portable manipulandum setup. So there's a tablet underneath and a screen on top where participants view the stimuli. And then there's a handle in between that holds the stylus that coordinates with the tablet and therefore the cursor on the screen. At UC Davis, we have the robotic manipulandum, the kin arm. And again, participants are looking down on a screen where the stimulus is being shown, and they have a handle underneath that they move around. So the study that was done by Rose DeCock in Will Joyner's lab, just to kind of test the basics of the framework of whether movement and auditory together are more accurate, more precise. So they had participants do a temporal reproduction task, and during the encoding phase, they were able to move the robotic arm around in the movement condition, whereas in the auditory condition, they had the arm was locked into place and they just heard the auditory tone. Whereas in the combined condition, they were able to move around while hearing the auditory tone. And then to reproduce, they simply pressed the button to start and stop, but pressed and hold the button to reproduce the time. And they found that the multisensory condition, the movement plus auditory condition, was more accurate than either unisensory condition alone. So it does appear that the addition of movement to auditory does improve the time perception. So to further verify the Q combination framework, we wanted to investigate the hypothesis that if movement becomes unreliable or noisy, we should see this decrease in time perception precision. So we wanted to make movement noisy and see what would happen. We hypothesized that the precision of the estimate will be pulled toward the more precise sensory input. So to test this, we had participants do a temporal bisection task. In one case, the temporal stimulus was an auditory tone, and in the other, it was a visual global background change. So the screen color would change for the duration and then change back. And we had, again, it's like basic temporal bisection test. The participants would move to a starting point, and then the auditory tone or the visual stimuli would start as they were moving around. Moving the robotic arm around. And then when the stimulus ended, they would move to one of two targets to indicate if the duration was short or long. But to add noise to the movement, to stimulate a tremor-like movement, we had 3 different levels of frequency and 3 different levels of amplitude that they could experience on any given trial. So frequency you can think of as the speed of the tremor, whereas amplitude you can think of as the size of the tremor. So for example, a low frequency trial would be a small, slow movement, whereas like a low amp high frequency would be a small, fast movement. A high amp high frequency would be a large, fast movement, where a high amp low frequency would be a large but slow movement, just to give you a kind of sense of what those different levels would look like. And there was 9 different combinations they could experience on any given trial. So just a quick video, you won't see the hand itself shake since it's artificial, but you can see the cursor itself shake. So participants would move to the starting point, an auditory tone would play, and then they would move to one of two targets to indicate short or long. So on to the results. I'll talk about the auditory results first. Repeated measures ANOVA found there's no significant difference on bisection points. So neither amplitude or frequency had an effect on the accuracy of timing. So we use this as saying, the tremor was not distracting to participants. They were able to still perform the bisection task. However, when we looked at the CV, we found that there was a significant effect of frequency, but not amplitude. However, post hoc analyses were only marginally significant. There does appear to be a linear effect here. So we explored that linear comparison of frequencies across all levels of amplitude and did find a significant linear effect of frequency on CV. So as frequency increases, CV increases, therefore precision is decreasing. As for the visual study, again, we did not find an effective bisection point. However, in this case, there was only a marginal effect of frequency, but not amplitude on the CV. But again, there appears to be this kind of linear relationship. And there was a significant linear effect of frequency on CV in the visual study as well. And that frequency increasing led to an increase in CV. But what's more interesting is comparing these two effects side by side. So the first thing you'll probably notice is visual CV is higher than the auditory, which would be expected given what we know about visual and auditory timing. But what's more interesting is kind of this reverse pattern that you see. So the frequency, as frequency increases to frequency 8, it kind of levels off between 8 and 12. Whereas in the visual, it stays level between 4 and 8 and then increases at 12. And we suggest that this is showing that as frequency increases, the auditory unisensory estimate becomes more precise than the movement unisensory estimate. And that's why we see this kind of leveling off. The movement information is no longer important because it's become too noisy. Therefore, it's just following the auditory estimate. Whereas in visual, because visual timing is less precise to begin with, movement remains still the more precise unisensory estimate until a frequency of 12. And we suspect that if we continued upping the frequency, we would still see a leveling off from 12 on. So just to kind of visualize how this relates to the Q combination framework, I have this kind of little video where you'll see the standard deviation of the movement unisensory estimate changing. So you can see the blue is either auditory or visual unisensory estimate. The dark orange is going to be the movement unisensory estimate, and then the lighter orange yellow is the multisensory estimate. So as the standard deviation gets larger, you can see the flattening out of those curves, and you'll see that the unisensory auditory or visual estimate is closest to the multisensory estimate. And we, as I said in the previous one, the auditory becomes the most precise at a lower frequency than visual. So at frequency 8, auditory becomes more precise than movement, whereas with the visual study, visual doesn't become more precise until the frequency of 12. So just to summarize, as we hypothesized and in line with the proposed Bayesian Q combination framework, noisy movements led to more variability in perceptual timing. And importantly, the temporal estimates were pulled toward the precision of the other sensory modality, more so for auditory than for visual. And interestingly, the speed of the tremor, but not the size of the tremor, had a greater effect on temporal encoding. So frequency, but not amplitude, had the effect. So in addition to looking at bisection point and CV, we also looked at some of the movement parameters. Specifically, we looked at movement length. So this is defined as the sum distance traveled during the encoding period, as well as movement force, which is the summed instantaneous force applied to the handle during the encoding period. So first we'll look at movement length. So in the auditory study, there was a marginally significant effect of frequency, but not amplitude. And in visual, there was a marginally significant effect of amplitude, but not frequency. And again, we suspect a kind of linear effect. So we explored that linear comparison. So for the auditory, we found movement length decreased as frequency increased, whereas in the visual, movement length increased as amplitude increased. As for movement force, we found significant effects of frequency, amplitude, as well as an interaction effect. So left is auditory, right is visual. So post-hoc analysis of the frequency, first you'll see that movement force was overall higher in visual compared to auditory, but overall movement force decreased with frequency in both versions of the experiment. So as frequency goes up, movement force goes down. As for amplitude, again, amplitude is overall higher in visual, movement force is overall higher in visual than in auditory. And overall movement force increases with amplitude in both auditory and visual versions of the experiment. So amplitude increases, movement force increases. And since they're working against each other, so frequency is decreasing and amplitude is increasing, we see these interaction effects. So movement force remains constant across all levels of amplitude for the highest frequency, but movement force overall increases as amplitude increases for the low and mid frequencies. So just to summarize these findings, so for movement length overall was longer in the visual compared to the auditory study. Specifically in the auditory, an increase in frequency led to a decrease in movement length, whereas in the visual, it was an increase in amplitude leading to an increase in movement length. We looked into whether or not movement length was affecting the accuracy, being that it's a different movement statistic and that would be expected, but there was no significant effect by section point. They were slightly different, but nothing significant. As for movement force, it overall appeared to be more force applied in the visual study compared to the auditory. Specifically, the increase in frequency led to a decrease in force, whereas an increase in amplitude led to an increase in force. And we suggest that this is just the nature of the beast with having to control the robotic hand. Participants needed to apply more force to control it at higher amplitudes. Because if they let go of the handle too much, it would lock into place and they would have to start over. So I think they were just applying more force to remain in control of the robotic arm. So just summarize overall, so in line with Bayesian Q combination framework, increasing the noise of movement decreased the precision of timing. And as for movement length and movement force, we didn't see the movement parameters actually having any effect on time perception itself, but suggest that there's actually an interaction effect going on here, that increasing frequency led to decreased movement force, which lead to decreased movement length, and an increase in amplitude led to an increase in movement force, which increased movement length. That's all I have. Thank you, guys.
Thank you, Keri. Let's go through the participant list to see if there are any questions coming up for your presentation.
Get everything back up here.
I can see clapping hands.
If you want to, I can't see everybody. If you want to call on people, that's fine.
Okay, you can see everybody.
I just, because I have the screen shared, I can't like scroll through easily.
Okay. Yes.
If you don't mind calling on people, that would be great.
Yeah, and you can also have a look at the chats button.
I see Jonathan. I'll let you go, Jonathan.
Yes.
Hi. Yeah, thank you for a really interesting presentation. I've been following this story, and it's fun to see this stage.
Awesome.
I guess I'm wondering why you think it might be the case that amplitude didn't make the same difference to precision as frequency did. Is there a reason that higher frequency should be noisier, whereas higher amplitudes not so much?
It could be. I mean, if you think about movements in general, right? Like we have different size movements all the time, but we often have like a speed tremor issue. So I think it might just be the nature of frequency versus amplitude. But yeah, it's unclear at the moment. But yeah, it's definitely one of those things. I think it's just the type of movement statistic that we're looking at as of speed versus size.
And then I guess one other quick question.
Sure.
Do you picture this as something that's a Bayesian judgment that's implemented at the decision point, or something that's continuously influencing the perception of time elapsing over the course of the movement?
Speculatively, I think it's over the course. I don't think it's at the decision process. I think it's the noise during the actual timing itself that's causing the influence.
Thanks.
Yeah, thank you. What else? Any other questions? I don't see any hands. Does anybody else?
Yeah, me neither.
Okay.
Last check to the chat. Yes, it was pretty clear, your presentation. So you explained everything very well, even to people that they don't do research specifically on motion. So I guess that is why. If you have questions during the whole journal club, we can definitely go back and answer them if we have the time.
Sure.
If that's okay with everyone. So let's move to our second talk for today. So the second speaker is Dr. Tomas Lenc. He is also a postdoctoral research fellow at the Rhythm and Brains Lab in Belgium. He completed his PhD at Mark's Institute for Brain Behavior and Development in Western Sydney University. And during his PhD, he used surface EEG and behavioral methods to clarify the nature of processes that support perception and sensory-motor synchronization with musical rhythm. Currently, he continues to follow this line of research, aiming to map the functional network of cortical regions involved in musical beat processing. He uses human intracranial EEG recording from patients with epilepsy. He's generally interested in the internal representations of rhythmic sequences, and particularly on how the brain maps from an infinite space of possible rhythmic sensory inputs to a limited number of perceptual categories. So the title of today's talk is Internal Representation of Musical Rhythm, Transformation from Sound to Periodic Beats. So, Thomas, the floor is yours.
Thank you. Can you hear me well?
Yes.
And you should be able to see my screen as well. Okay.
Yes, we do.
Excellent. Yeah, so hi, everybody. Thank you again so much for the opportunity to discuss this quite exciting data with all of you. And my name is Tomas. I'm a postdoc in the lab of Sylvie Nozaradan in Brussels. And today I will talk about this line of work that we have been developing over several years. And the overarching idea is to use musical rhythm as a particularly useful way to better understand the nature of time and coordination in humans. So why music? Well, in musical scenarios, humans across all cultures show a remarkable capacity to tightly coordinate movement in time. So we can have musicians, dancers, or simply listeners clapping and bobbing their head in time with music and with each other. And these coordinated behaviors are typically based on a particular kind of internal time reference called the beat. So beat is a perceptual representation of a periodic pulse, here in orange, that is elicited by a rhythmic sensory stimulus, here in black. And periodic pulse simply means a series of regularly recurring points in time that can be used as an internal time reference to produce precisely timed movement. And now the key thing is that the perceived beat is not necessarily one-to-one with the arrangement of stimulus acoustic features over time. So in some cases, rhythms can be arranged in a way that is very close to the elicited beat, such as in this example where the tones in black tend to systematically overlap with the time points of the typically perceived beats in orange. So this rhythm can be called a strongly periodic because it has a recurrent acoustic structure that provides a direct cue from which the beat can be essentially extracted. But it gets even more interesting. For example, we have a stimulus that does not align very well with any periodic pulse, and hence we can call it a weakly periodic rhythm. Still, this stimulus elicits a consistent perception of a beat, showing that our perception goes far beyond the acoustics. So in other words, rhythm perception is not just about faithfully encoding the temporal structure of stimulus features, but it's about mapping from stimulus onto a categorical internal template, in this case, the beat. And so our goal is to try to understand the nature of this categorization. So to do that, I will first show you the work we did at Western Sydney, trying to capture this mapping using surface EEG. So EEG is a technique that measures synchronized activity of large neuronal populations from the scalp surface, and it has a very good temporal resolution. So here is the paradigm we developed. We would take a stimulus based on a repeating rhythmic pattern of tones. Now, this is a strongly periodic rhythm, so the tones are arranged to closely align with a periodic pulse. When we extract acoustic modulations in the stimulus and transform that into the frequency domain, we get a series of peaks, which represent how the energy in the rhythmic sound fluctuations is distributed across different frequencies. Now, here is the EEG response that we recorded from the scalp surface of 14 participants as they were listening to the rhythm without any movement. Now you can see peaks at the same frequencies as in the stimulus spectra just above that represent activity elicited by the rhythm. Now, here in red is a set of beat pulses most consistently perceived by participants when they were asked to tap along the rhythm in a separate session. And you might ask, why is there not just a single beat, but a multiple? Well, in fact, people often perceive a whole nested set of pulses simultaneously when listening to musical rhythms, and this set is called the meter. So here we will simply consider all the beat pulses from this perceived meter at once. So based on the periods of these perceived pulses, we can tag frequencies in the sound modulation spectrum as beat related in red, corresponding to the frequencies and harmonics of the perceived beats. And the rest of the peaks are tagged as beat unrelated and shown in blue. Then we z-score the amplitudes across all peaks, and we take the mean z-score at the beat-related frequencies, the red ones. So this tells us how much the beat-related frequencies stand out in the spectrum relative to beat unrelated frequencies. And we see that the z-score taken from this stimulus spectrum is high, meaning that the beat frequencies stand out a lot. So in other words, the perceived beats seem prominently represented in the physical structure of this strongly periodic stimulus, which is what we would expect. Now, here in purple are the EEG responses across individual participants. On top, you can see a distribution of the average magnitude at beat-related frequencies across all the EEG sensors. So just simply imagine looking at the participant's head from the top down, and then the brighter the color, the larger the magnitude at beat-related frequencies that was recorded at that scalp location. So overall, we see that the beat-related frequencies are very prominent, similar to the stimulus. But of course, in this case, the response can be trivially explained by simply passive neural tracking of stimulus modulations. But let's say we take a stimulus that is acoustically far from a periodic beat, a weakly periodic rhythm. And what's cool is that this rhythm also induces perception of a stable set of beats, as we can see when we ask participants to tap along. However, notice that now the frequencies where participants consistently perceived beats are not very prominent in the acoustic structure of this rhythm. So what do you think we're gonna observe from the brain? Well, we see that beat-related frequencies are actually enhanced in the EEG response, even though this cannot be directly explained by the sensory input. So instead of tracking the stimulus, the brain seems to selectively enhance the representation of periodicities where participants tend to perceive beat pulses. So this first study showed us that the mapping from sensory inputs to beats is associated with a neural transformation that renders stimuli more periodic than they are in reality. And so one interesting question is whether this capacity to transform weakly periodic rhythms emerges early in development, or whether it requires prolonged exposure or maturation. And so to address this, we used our approach to analyze EEG responses of 5 to 6 month old infants. They were passively listening to the weekly periodic rhythm. And so this work was done in collaboration with the Baby Lab at Western Sydney University. Now, because infants cannot tap the beat, we instead tagged beat-related frequencies based on the previous tapping data from adults. Now, in gray, you can see the stimulus waveform, and in black is a simulated response from the auditory nerve. And from now on, I will call this a cochlear model. And so we will use this simulation to test whether the neural transformation observed in the EEG could be explained by early nonlinearities in the auditory system, or whether it emerges from some higher level, perhaps cortical processing. So here is the spectrum of this cochlear model. Notice again that the beat-related frequencies don't stand out in the spectrum, so their mean Z-score is rather low. And here are the EEG, and here are the corresponding EEG responses averaged across 20 infants. And just as we saw in adults, the responses across infants have more prominent beat frequencies than could be expected from the cochlear model alone. So we see that neural processes which kind of periodize the representation of weakly periodic stimuli are in place early in development. In fact, this is way before the ability to synchronize overt body movement with music develops. And our cochlear simulations also show that the transformation is unlikely to emerge from early auditory nonlinearities. So where in the brain does the transformation emerge? Well, to answer that question, we need more spatial resolution than surface EEG can offer. And to achieve this spatial resolution, we developed a collaboration with Bruno Rossillon at the hospital in Nancy. And with them, we collected data from human patients undergoing clinical monitoring for treatment of medically refractory epilepsy. So these patients were implanted with stereotactic EEG for clinical purposes. And while they were staying at the hospital, they volunteered to participate in our experiment. So SEEG is a method that records electrical brain activity intracranially. The way it works is that these narrow electrode shafts are inserted by the neurosurgeon to penetrate brain tissue through small holes in the skull. Each electrode shaft has from 5 to 15 independent recording contacts, and the exact location of the implantation is always determined only by the clinical needs. So here are all the electrode contacts merged across 13 patients, and participants listened to the same weekly periodic rhythm as we used in the studies before. So first we selected contacts generally responsive to sounds, ones with clear spectral peaks at frequencies expected from the stimulus. And then we looked at the transformation towards periodic beat only within these generally sound responsive contacts. So here, the more red the contact, the more prominent beat-related frequencies are in its spectrum. So just to go one step at a time, we first wanted to test whether there was a significant transformation already at the level of the primary auditory cortex. So we used this combined anatomical and functional localizer to isolate contacts from the primary auditory cortex. And again, we have the stimulus in gray and simulated auditory nerve response in black, its spectrum just below, and a low prominence of beat-related frequencies in this simulated peripheral auditory response. And here is the average response from the primary auditory cortex during passive listening. And as we can see on the right, the beat-related frequencies are selectively enhanced compared to the cochlear model. So indeed, the transformation emerges quite early, very early at the cortical level. So what about regions beyond the primary auditory cortex? Well, taking only the sound responsive electrodes, we observe that beat frequencies are selectively enhanced in a number of perisylvian regions. This includes the primary auditory cortex, as we saw before, but also parietal sensory motor association areas, particularly the supramarginal gyrus. And this is actually very interesting because, for example, studies of speech processing show that parietal cortex links auditory and motor systems and is heavily involved in sensory motor transformations. And we have also confirmed these findings using an alternative analysis method based on autocorrelation that has some really cool properties to complement the typical frequency tagging approach. And I'm just going to leave this as a teaser, and feel free to ask me in the discussion if you're interested to hear more. So we can add to our summary that the transformation of weakly periodic rhythms is in place already in early auditory cortex. And so this leaves us wonder whether we are capturing some kind of basic cortical mechanism that we may even share with other primates. And to test this, we developed a collaboration with Hugo Merchant's lab, and they recorded the responses from the macaque auditory cortex as the monkey was passively listening to the weakly periodic rhythm. And so the recording electrode had 64 contacts along a linear shaft which penetrated the cortex, and we analyzed local field potentials only from sound responsive contacts. So same as before, we have stimulus in gray, its amplitude envelope in black, sound envelope spectrum tagged according to human tapping data, and a low beat Z score taken from this spectrum. And here is the brain response averaged across all the rhythm cycles, all responsive electrodes and recording sessions. And on the right, you can see data from the 39 recording sessions shown as purple points. And the pattern is quite clear. We see something very similar to humans. In other words, even monkey auditory cortex doesn't simply just track the envelope of this weakly periodic rhythm, but somehow selectively enhances representation of the beat that humans tend to tap. Okay, so this is quite interesting. We have a brain mechanism that is present in human adults, but also infants and monkeys who both cannot move to the beat. And in human adults, this mechanism emerges early in the primary auditory cortex. So putting it all together, it seems like there is this fundamental early auditory cortical processing stage, which renders rhythmic inputs more temporally recurrent or periodic than they are in reality. And so this general mechanism outputs a periodized default format of the input, which then can be exploited by higher level associative and motor areas to coordinate movement to music. And we expect this would depend on the quality of functional coupling between the auditory and motor systems, which is weak in monkeys and presumably infants. And there may also be individual differences in human adults, for example, due to musical training, etc. So together, our data seems to highlight one of many neural processes that together underlie coordinated musical behaviors observed across human individuals. And so that's just to give you a taste of some things we are working on in the lab right now. For instance, in all these studies I've just presented to you, we looked at kind of general fixed mechanism that shapes rhythm representation. So the next step is to look at the flexibility and dynamics of the neural transformation at different timescales, from lifetime to just the last few seconds within a musical piece. And we are also trying to generalize the findings beyond this one weekly periodic rhythm. And we have also this new exciting project that uses representational similarity analysis to capture rhythm categorization when sounds are shifted on a very fine time scale. So that's it. Thank you so much for listening, and I'm looking forward to the discussion.
Thank you so much for presenting your evidence. We already have raised a hand. Let me go through the list to avoid any mistakes. It's Chris Paschalidis.
Yes, hello. Hello.
Hi, Chris.
Can you hear me?
Mm-hmm.
Yes.
Okay, so given that the transformation you told us about happens as early as the cochlea, does your results align or do not align with NRT neural resonance theory for beat perception, or what does it say about you? In the end, you said about the motor cortex, but in the beginning of the presentation, and I didn't get the connection.
No, thank you. That's a question. So actually, just to clarify, it's not the cochlea where the transformation happens. It's actually higher. So we observed it in the primary auditory cortex, and it seems like that already at that level, the beat is selectively enhanced compared to what we see when we simulate responses from the cochlea. And that's especially for these rhythms where you don't have prominent beat representation in the sound already. And in terms of neural resonance theory, yeah, I think these results actually go quite well with the models and the work that's been proposed before by at large, talking about some general mechanisms. They explain it by neural resonance. So we just provide the data. We don't talk about the mechanistic explanation, but this goes quite well with it. So you have some basic mechanism based on perhaps the network, but perhaps it's also learned. I mean, it emerges quite early in infants. So yeah, definitely in line with the neural resonance theory.
Thank you.
We have another question from Jonathan.
Hi, yeah, I'm excited to see all of this work. Is based on the fact that you're seeing it in primary auditory cortex, that suggests that it's emerging early in the processing pathway. But is there anything in your observations that would preclude this beat-related activity in primary auditory cortex coming from somewhere else, for instance, motor areas?
Yeah, that's an excellent question, because I tend to think about the auditory system and brain in general as a network rather than a feed-forward hierarchical processing device. So yeah, the fact that we do observe it in cortex, in the primary auditory cortex, does not mean that the shaping of the response doesn't come from a larger network of interconnected areas. So yeah, I would definitely not exclude the fact that this is due to connections to other regions.
But I guess in that one experiment, you had such extensive brain coverage, you could at least maybe narrow down and say like, well, it can't be coming from here because there aren't beat-related enhancements there. But I guess you saw the beat-related enhancements in a lot of different areas. Yeah.
So that's the thing. we did have electrodes implanted in some motor regions. We did not have that many in, for example, the dorsal premotor cortex. We had some in the SMA. I didn't show the data, but it was not very convincing. So from the regions where we had implantations, for example, in the inferior frontal gyrus, there was a significant enhancement of the beat representation compared to the cochlear model, but actually we saw the sharpest representation in the auditory cortex and in the supramarginal gyrus. So yeah, it seems that it's really these early regions that have the sharpest, most transformed representation. And then perhaps, I mean, as I said at the end, this was a passive listening task. These were human patients who actually could not really tap the beat so well. So what could be happening is that we have this basic kind of transformation in the in general transformation in the auditory regions and in the parietal regions that are part of the dorsal stream. But then whether or not this is completely transferred and perhaps even more sharpened in the frontal regions and the motor regions may depend on the individual differences, but also on the task. So yeah, I guess that would be my question. So I tend to think of this as kind of like basic format that is there that might be biasing what happens with it afterwards, and whether it gets further used and shaped by these frontal associative areas that might depend on the connection and the task and what's actually going on. I guess when you ask people who study monkeys, they will tell you that these frontal regions with a passive task that you will not record anything. So anyway, no, but thank you for the question.
Thank you.
Now there's a question from Erica.
Yes, in the chat.
Would it be possible to determine the pathways by doing time frequency decomposition in each of these areas where the electrodes are to determine the order in which the various beat-related frequencies are processed? Thanks for the question, Erica. So you're saying that we can do time frequency decomposition in each area to determine the order. So I guess, well, so the beat frequencies that we tagged are like the harmonics of the different beats that are perceived. We ask participants to tap, and they are kind of quite consistent in terms of the period of the beats they tap, but they may be choosing like multiples. So, you know, the beat and then twice as fast or 4 times as fast. So that's the meter. And when we tag the beat related frequencies, we take them as a whole because all the harmonics actually in the spectrum, they overlap and they together describe the prominence of the periodicity. So taking each individual harmonic in the spectrum, that is not necessarily, it doesn't necessarily tell you much. What we did before, for example, was using time frequency decomposition to, and this is work Sylvie has done with surface EEG, to isolate responses from the subcortical regions. That was done. That was done in a paper from 2018, and that can be done. I can show you the slides actually, because we did something similar. So, for example, you can play the rhythm with a complex tone, using a complex tone. So this is a representation of one cycle of the rhythm, and then this complex tone that is playing the rhythm has 3 partials. They are not harmonics of each other. So where you look at the spiking in the auditory nerve and take the FFT, you see that you have all these distortion products that are actually centered at these very high frequencies that are like the frequencies of the tone. So for example, this is one of the distortion products that is at 168 Hertz. Now this frequency is very high. So actually the beyond primary auditory cortex, the neural populations are not capable of tracking such high frequency. And now you have all these sidebands that tell you the modulations of the neural responses that do track this very high frequency. So you can use this to learn about what is the shape of the response that is tracking this very high frequency, and you can look at whether there is a transformation there, and then you can look at the envelope following responses, which are low in the spectrum, and this will give you the idea of the whole network if you're doing surface EEG. And so this is one way to kind of decompose more spatially what is going on. So for example, that study that I mentioned, the 2018 study from Silvino Zaradan, we didn't find selective transformation or enhancement of beat frequencies in these subcortical, putatively subcortical responses, while we still did find them in the envelope following response. So yeah, this can be done, but in terms of looking at across different regions with intracranial EEG, this is a little bit tricky. Yeah, you can look at the phase differences, but then this is yeah, one has to be quite careful because the shape of the response can differ between two regions, and so just looking at the phase might simply be, there might be simply phase differences due to the shape of the response rather than actual like latency differences. I don't know if this long answer answers the question. We can chat more, Erica, as well at if you're at LPPW or in October at TRF.
He says, sounds good. I think the question is covered. Nice. If there are no more questions for Tomas, I think we can wrap it up here. There were really interesting discussions held today, and thank you for giving these two interesting presentations. We will see you hopefully in TRF. Most of us will be there. So we will have another journal club for this academic year at the end of June, approximately. We will share the information as soon as we have them. And we will continue the journal clubs from September on again. Thank you, everybody, for attending. And again, thank you to the speakers for accepting the invitation.
Thanks for having me.
Thank you so much.
Bye, everyone.
Bye.